Okay, so after you filled out the Punnett square, you should get something like this. You have a homozygous dominant individual, you have 2 heterozygotes, and then you have 1 homozygous recessive individual. This is showing all the possible genotypes that the offspring could be. Now, we talked about the difference between genotype and phenotype, so you need to make sure that you look at the difference in ratios between the two. So if we talk first about the genotypic ratio, which I'm just going to put a G here. So the genotypic ratio is going to be the ratio between all of the different genotypes that are possible in the offspring. So if you look at this one, you have 3 different possibilities. You have this as 1, the homozygous dominant. This is 2, so your heterozygous individual, and then 3, your homozygous recessive individual. So if you look at that, you need to write a genotypic ratio for that. So if we do that, you would have something like this. You would have 1 big R individual. You would have 2 heterozygous individuals, and you'd have 1 homozygous recessive individual. Okay? Now, the difference between this and your phenotypic ratio is that in the homozygous dominant and the heterozygous, their phenotype is going to be exactly the same. And so that's something that you have to consider, because then that makes the ratios different between the genotype and the phenotype. So we'll put a P here for the phenotypic ratio. And if we count, and we have to look at what our original thing said, so we said that round was big R and wrinkled was little r. For it to be considered round, it only has to have 1 big R, because we said that up here, dominant, it masks the recessive allele with only 1 present. So this would be 1 individual that's round, this would be another, and this would be the third. So the phenotypic ratio would be 3 round to 1 here, which is the homozygous recessive, and they would be the 1 wrinkled seed. And then you can answer questions about this. How many individuals are heterozygous? 1, 2, 2 out of 4, 50%. How many individuals are homozygous recessive? 1, so that's 25%. How many seeds would be round? We said that here, there's 3 out of 4, which would be 75%. So that's essentially how you do monohybrid crosses and how you interpret the data that's in a Punnett square. So my next workshop will be talking about dihybrid crosses, which is where you look at more than one trait.